Hi guys, how are y'all doing today? I'm doing wonderful. I'm making this on a Friday. So this week we talked about our scientist and how we got to where we are with DNA. So now let's learn a little more about it. We're just going to briefly go over the scientist one more time, the history of heredity. And we're going to start with Gregor Mendel. Now, Gregor Mendel was the one who did the pea plants, and what he found out was that genes are passed from generation to generation. He made the laws of inheritance. And remember, we talked about in class how before this, people thought, oh, if I have a white flower and a red flower, all my flowers will be pink from those. Well, that's not right. And he proved it. He proved that there are dominant and recessive genes and that they can change how many offspring look different. So that's why he was important, because he got everyone interested in genes and how they affected her heredity. So Frederick Griffith is our next guy. He's the one who put the bacteria in the mouse. So there was a disease-causing bacteria and a harmless bacteria. So clearly the disease one killed the mouse. The harmless one did not. But then when they killed the bacteria using heat, the mouse lived. Yay! But oh wait, they put that heat-killed disease-causing bacteria with the harmless, and it killed the mouse. So he was like, what? How could that happen? So he was like, you know what? That's the transforming principle. That's what we're going to call it. We're going to call it the transforming principle because something obviously changed in there. But really, what was that? What do we think that was? The transforming principle that allowed this dead bacteria and this harmless bacteria to still kill this mouse. Well, it's DNA, which is what we're talking about in this entire unit. So this is a molecule of DNA, and it is made up of nucleotides, which are the monomers. And it has it helps us store our genetic information and make proteins. So after Frederick Griffith said, hey, that's the transformation principle, Oswald Avery came in and he was like, that's actually DNA. And he determined that genes are composed of DNA, not just protein. Then we have Erwin Chargaff. Erwin Chargaff is super important because he analyzed the nitrogenous bases, which are adenine, thymine, cytosine, and guanine. And he said, it's really weird that adenine and thymine have the same numbers, and so do cytosine and guanine. They must pair together because why else would they have the same numbers, right? So those are the pairs, which we will get more into in a second. Rosalind Franklin and Maurice Wilkins, they were super awesome, and they used X-ray crystallography to take a picture of DNA. And this very famous photo called Photo 51 is the first proof that DNA was a double helix. So after they did that and got that picture, Watson and Crick came around and they decided to try to work on building a structural model of DNA. So that's what they're posing with in this picture here, is their model that they created. So if you have a second, you don't have to write this in, but I want you to at least think about it. What is one word that describes the contribution that these scientists had towards DNA? You can pause this video if you want to take a minute to think about it or write it in. So let's talk about structure. So like I said before, the monomer for DNA is a nucleotide. Now a nucleotide has three important parts. It has its phosphate group right here, the sugar, and the nitrogen base. Now this sugar right here for DNA is deoxyribose sugar, okay? So those are the three parts that make up the nucleotide. Here's our DNA, and these red boxes are supposed to symbolize a ladder, because if you lay DNA flat, we know it's kind of like a spinny, swirly structure, but if you unfold it and lay it flat, it looks like a ladder. So here we have the stands of the ladder. I'm not sure what the actual word is, but I want you to notice what makes this up. Okay, so I'm going to go back to our picture really quick, and I want you to see what makes these up right here. It's the sugar and phosphate. So the sugar and phosphate are right here, and then what are those connected to? The nitrogenous base, which is either the adenine, thymine, cytosine, or guanine. And I want you to see in the middle that those nitrogenous bases connect to each other with these white lines, and those white lines represent hydrogen bonds in between the bases. So let's kind of zoom out of our DNA mindset for a second. So when we did our cell structure unit, we know that we have our nucleus, and from our nucleus, we have our chromosomes. From our chromosomes, we go to our genes, and then we have the backbone, our DNA backbone, and then the nucleotide. So that's kind of our level of organization for our DNA. So back to our base pairs, we have two different types, purines and pyrimidines. So adenine and guanine are our purines, and thymine and cytosine are our pyrimidines. One way you can remember this is that thymine and cytosine have a Y in them, and so does pyrimidines. Okay? And like we said earlier, thanks to Chargaff's discoveries, the A right here and the T are going to bind together, and so are the G and the C. Notice how it's one purine and one pyrimidine bonding together. The way I remember the correct pairing is that C and G look the same, and then A and T are left over, so they have to bond to each other. So again, here we have our little ladder structure. We have our phosphate group, our sugar, and then our nitrogenous base. So here's our C and G's. Here's our T's and A's. And notice it doesn't matter what side it's on, it just has to be bonded to that other nitrogenous base that it matches with. So let's talk about why we have DNA. What do you think? I want you to kind of pause this video for a second and think about why DNA is important to us. What does DNA code for? What actually makes you look the way you do? DNA or proteins? So for the first question, why do we have DNA? Well, DNA contains our genetic code. It's our genetic makeup. It's what makes us us. Like, for what does it code? It codes for proteins, okay? And then what makes you look the way you do, DNA or proteins? It's going to be the proteins that make you look the way you do. Now, I did write notes on this, so I want you to make sure you look at those and copy them down in your notebook, because like I have said many times before, it's going to really help you out on your test day to have those notes right in front of you and not have to have like 40 tabs open. And the last thing I want you to do as you're writing these notes is I want you to practice making a complementary strand. So a complementary strand is looking at the nitrogenous bases. So on this left strand, we have A, C, T, T, etc. And for the right strand, I want you to remember the base pairing rules. So A goes with T and C goes with G. So let's do the first three letters together and then try the rest on your own. So if this one is A, my first one on the right strand has to be T. My second one is C, and C and G are base pairs, so it's going to be G. My third letter is T, and A and T are base pairs, so it will be A. So why don't you go ahead and try to finish the rest of those on your own, and then check your work with the notes. do I stop the video?
No. Did you hit stop sharing?
No, I hit hide.